every day. The fact we can sit here learning, that's good, right? Yeah. So if you guys don't have additional questions, we can start looking at the first group of microbial infection. We're going to look at the bacteria, right? So first group bacteria, right? So let's review bacteria. You guys remember bacteria made of what type of cell? Bacteria. Gram positive, gram negative means this different cell wall. Yes. So gram reaction give you the information about the bacteria cell wall. So bacteria.
Your microphone's not on.
It's on, right?
I can't.
It is on.
It is? Okay.
It's on. Okay.
There we go.
Okay, good. So bacteria made of what type of cell? Prokaryotes or eukaryotes?
Eukaryote. Eukaryote.
Bacteria made of what type of cell?
Prokaryote.
Okay, this time we reviewed. Yeah. Bacteria. Good time we reviewed. Yes. Bacteria made of what type of cell? Prokaryotic cell. Prokaryotic cell do not have nucleus. Yes. Okay. Awesome. Good. Get that out of the way. Yes. Bacteria made of prokaryotic cell. Bacteria typically have cell membrane. You have the chromosome DNA in the nucleus region. You have ribosome, plasmid. Some have cell wall. Based on cell wall, based on gram stem, we have gram positive, gram negative bacterial cell wall. Some bacteria also have additional lipid on the gram positive bacterial cell wall. We call them acid-fast positive bacteria. And some of them also have capsule, fibria, pili, flagella. So we learned that in microbiology 1. Hopefully you guys still remember it. Yes, of course. Awesome. So that's review of bacteria. Right? Now, let's look at nervous system. So nervous system, some of you guys said, oh, we learned some nervous system in our class, right? First sentence, can you find a normal microbiota in human nervous system? No. Yes. No. So if there's microbes, any type of microbes in the nervous system, that's disease, yes? Okay. So the central nervous system include what? Central nervous system, brain and spinal cord. Yes? And so the infection in the brain, we call them what? Encephalitis. Okay, good job. There you go, right? Awesome. And then the peripheral nerve. Peripheral nerve. See, this is a picture to show you the peripheral nerve. Yes? Long concord nerves. Yes. So we can sense in the sensing, touch, eat, all that stuff. Yes? Okay. Now, this is a good review. So you guys learned the neuron, the cell, right? The neurons made of what type of cell? Prokaryotes or eukaryotes? Eukaryotes. Oh, good. And then we have list of bacterial infection, common virus, the protozoa, fungi, worm infection, prions. We're going to look at all this. Okay? So this is really good review. Okay? So now we're going to first sort out the bacterial infection. But which structure allow offer the cushion and the nutrients, nourish the spinal cord, nourish the brain? Yeah, cerebrospinal fluid, right? So if you have a cerebrospinal fluid, you see the membranes, pia mater, dura mater, erythroid mater. You guys all learn that? Yes, 3 membranes. Yes. Have you learned that? Yes. Okay. You learned that. So if your microbes in the cerebrospinal fluid cause inflammation, this is the membrane. What do we call it? What's the name of the disease? Meningitis. Yes, meningitis. So the encephalitis, meningitis, right? So if somebody have meningitis, how do we diagnose? How do we diagnose the meningitis? Spinal tap, right? You see the top, show you the spinal tap, right? You can grab the fluid, right? Through spinal tap, the cerebrospinal fluid. Once you get the sample from the spinal tap procedure, what do you do with it? Analyze it. Yes. So you can see it, right? Based on what type of regular cell, based on the glucose content, right? And the protein, you can kind of figure out if it's a bacterial meningitis, viral meningitis, or fungal meningitis. Yes? Right? So let's go. Since this first group of disease, we're going to look at bacterial meningitis. after eating. Yes. If it's viral meningitis, what happens to the glucose level? That would be normal because virus cannot eat the glucose directly. Yes? Make sense? Okay, because virus is not composed of any cell. Okay, awesome. And then the vital cell, you see, if it's a polymorph nucleus, polymorph nuclei neutrophil, so that's bacterial meningitis. So the neutrophil increase associated with what? Bacteria, right? And other lymphocyte increasing indicate what? Viral infection. Make sense? So you can analyze the sample based on the sample, the cerebrospinal fluid analysis. You can kind of sort out what happened with it. Right? Plus, you can obviously look at under a microscope. Yes. So it's bacteria. You can see the bacteria under a microscope. Yes. Okay. Now, what are the common bacterial meningitis? So here's some overview. Okay. So Haemophilus influenza. We learned a lot of disease we learned in the last quarter in the chapter of disease in the respiratory system. You guys still remember disease in the respiratory system? Yes. So the pneumonia, for example. So if you have inflammation in the lung, pneumonia, because in the lung there's a lot of blood vessels. So some microorganisms, when they cause pneumonia, they could continue spread across blood-brain barrier. That's your meningitis. That makes sense? So, for example, Haemophilus influenza and Streptococcus pneumonia. Both of these bacteria we discussed last quarter. So they typically cause what? Bacterial pneumonia, and then leads to bacterial.
Which one is deadly?
They both deadly. That's why both of them vaccines potential. So meningitis can be deadly. Bacterial meningitis is more deadly than the viral. Yes. They're so deadly, we have vaccine. Make sense? Okay. And then the severe meningitis is we're going to look at today. And then Streptococcus agalactia. This is a disease we're talking about in the last quarter, in the chapter of disease in the urinary system and the reproductive system. Streptococcus agalactia is group B Streptococcus. So maybe affect blood, pregnant woman. And then, like if a baby passing through birth canal, they could have sepsemia or vacuum. So if you want to review Streptococcus agalactia, you can check it out. Chapter is over in the urinary system. Yes? The seromonas cautioners. So this is disease talking about the associated with food, right? Cold cut, right? So this one is a pregnant woman eat cold cut, the baby could have bacterial meningitis. We're talking about that in digestive system chapter. But anyway, so some of them you can go back to review it. But so all these diseases, for example, Streptococcus agalactia and the seromonas cautioners, who's at risk for bacterial meningitis? Trichus or pneumonitis, right? And then meningococcal meningitis, Neisseria meningitidis, who's at risk is people living in the college dorm or in the military camp. And Haemophilus influenza, Streptococcus pneumoniae, who's at risk? The children. So that's both of these are common childhood vaccine. Okay, to prevent that. Make sense? So different bacterial infection have a different at-risk population, different. Yeah. Any question about this slide? Okay. Right. So now let's look at meningococcal meningitis. So this is the bacterial causing it called Neisseria meningitidis. So Neisseria meningitidis, let's describe in the gram reaction. Neisseria meningitidis via gram, negative shape, Diplococcus, right? The arrangement of Diplococcus shape is the cocci, yes? And we have beautiful picture right here. Yeah. They have capsule, they have fibria, how to attach. And how do the stem cells immune response? They also produce what? IgA protein. inhalation, they can directly go to the nervous system, plus meningitis. Okay, so let's look at the port of entry. See the port of entry. Okay. Right. So this is gram-negative bacteria. So the inhalation and then bacteria multiply because they have multiple mechanisms to escape toxin response. You have bacteremia. When you have bacteremia, and this is gram-negative bacteria, then the endotoxin release. Endotoxin release leads to what? Decimated intravascular coagulation. When you have decimated intravascular coagulation, then you not only have symptoms associated with meningitis, high fever, stiff neck, pain, all that stuff, you also have a decimated intravascular coagulation, talking about the hemorrhage on the blood vessel on the skin, petechia. High fever, petechia, stiff neck, yes, that's a classic symptom of meningococcal meningitis. Right? So it's several of toxins involved, and that's why there's vaccine available. And then the at-risk population is young adults. Oh, we're going to go to college, live in the dorm, or I'll join the military, living in the camp. Those type of crowded situation. Because when you live in crowded situation, the droplet, inhalation, droplet transmission, yeah. And this is gram-negative bacterial infection. Once you diagnose with the antibacterial antibiotics, can be easily penicillin, ceftriaxone, medicine, vaccine, all good. Now, you see, right? So this is a review of meningococcal meningitis inhalation, and then you through the olfactory nerve, you can get to the access to nervous system. Inhalation, bacterial access blood vessel, because they have a mechanism of capsule, escape phagocytosis, proteins, digest IgA proteins, digest the antibody, so they can easily become sepsemia, bacteremia, so then you have skin suffering. Right? You see the petechia? Right? So it have bacterial effects on which white blood cell increase. Which white blood cell increase in response to bacterial infection? Neutrophil. So what's a neutrophil look like? See right here. So if somebody have meningococcal meningitis, you do the cerebrospinal fluid, and what you see the Staphylococcus plus log. Right? And then if this one, like for the babies, right? For the babies acquires meningococcal meningitis, you don't have what? Sepsis. So prevention for this, which doctor can identify meningococcal meningitis, right? In terms of petechia or meningitis. Which doctor? Chiropractic doctor, you can measure body temperature, high fever. And you say, hey, doc, can you adjust my neck? I can't rotate the neck. When you're adjusting, oh, you have high fever. Probably it's not enough to just do the adjustment, but you have to get this meningitis treated, yes? Including, oh, you have petechia, purple dots. You see?
So what happens if somebody with meningitis comes in and like they just have to
The one thing is that it's inhalation, you need to wear a mask. Right? Yeah.
So sterilizing
Yes. So meningitis serious, you need to wear a mask to protect yourself, right? This once you like you're trying to adjust them, but they have stiff neck and have high fever, you might want to get them to the right? Why is that? Because people living in this country, one sense, they do not have access to medication. So if somebody gets disease, it's just, spreading. Because, and then also they live in a crowded situation, lack access to treatment, so they can spread. Enough. Right. Another one is Haemophilus influenza. So this is the where do you find Haemophilus influenza bacteria? They are very fastidious. Where do you find it? Not on the desk, not on your environment. Human carry. They are so fastidious they cannot grow on the blood agar. What type of agar they can grow? Chocolate agar. You have nice red blood cells. You can grow. So how to kill? Typically, which group of people are at risk for Haemophilus influenza infection? Typically, children. Because the moment, by the time you become a woman, you already had infection, you have immunity. So you carry this one by the organs. So the baby, how do they get it? From caretakers. Nanny, parents. So initial symptom with the Haemophilus influenza. This is gram-negative bacteria, the fastidious, human carrier, and the type B. This is most frequently associated with childhood pneumonia, leads to bacterial meningitis. So since bacterial meningitis can be very deadly because gram-negative bacteria also release endotoxin, so it can be very serious for the kids. So that's why they have this vaccine for children. Yeah. So how do we prevent Haemophilus influenza type B? Vaccination. Yeah. Now, so over here is summary, right? So streptococcus pneumonia, Haemophilus influenza type B, Neisseria meningitis all have vaccine because bacterial meningitis causes a lot of inflammation and injury can be very deadly. Okay. Now, nowadays, if you refuse to get vaccinated, you don't get vaccinated, right? You don't have to die for bacterial vaccine. Guess what? We have all kind of antibacterial antibiotics, right? So if you look at the drawing of the cell, look at this cell. This is what type of cell? Prokaryotes or eukaryotes? This is a prokaryotic cell. In the middle, because you don't have nucleus, yes, right? Prokaryotic cell. So this next couple of slides, including this one, is review of antibacterial antibiotics. Yeah, that's
If we already know, like, let's say if I didn't want to get vaccinated,
yeah.
and I lived in somewhere where it's hurting me, would I be like safe from that infection?
Yes, if you're not vaccinated, if you live in the area have disease, yeah, you won't get it. For example, which disease?
I'm saying like it said in an area where most people are vaccinated for that.
Oh, that's herd immunity. So if you refuse to get vaccinated, especially kids in America, so if you not get vaccinated, unvaccinated kids in America, less likely to get infection. Unless there's a diesel outbreak, then you could get the infection, right? So because in America, most kids get vaccinated, especially if kids go to public school, you have to use vaccines, right? So that unvaccinated kids in America get protected through herd immunity. But if the kids go to, hey, summer vacation, I'm going to travel to our area, right? And over there, they do not have herd immunity. They don't even have access to medication. Guess what? That kid will get sick. Make sense? Good question. Yes. So if you have chiropractic doctor, if you have a patient that carries baby come in, oh, we don't like vaccine because all the side effects, which is true, right? Side effects. So that you don't want to risk it. As long as you live in the community area, you don't give the kids pretty much safe. Yes, because herd. But if we say, oh, what are you going to do? I'm going to take my kids summer vacation to some area, right? Then, oh, you do not have vaccination. You can recommend the parents either get the kids vaccinated before you go, or take the medication because, yeah, right? Because you go to travel over there, you might not have access to medication. You see? So over here is a review of prokaryotic cell on the mode of target on what? Peptidoglycan. Could target on what? Because they do not have nucleus, the DNA RNA polymerase is unique, yes, could target on DNA RNA, the process of that genetic information flow. You can also target on cell membrane. If target on cell membrane in general, you need to use topic. Yes, we could also target on the ribosome, 70S ribosome, in prokaryotic cell, or the unique characters. Right? So this is a review of what we learned in last quarter about antibacterial antibiotics. Look at how many of those. Right? So when you go to the area, there's unvaccinated kids. They go to the area, there's you do not have that access to medication. If you do not want to get vaccinated, you just make sure you take medication with them. Yes? Okay. And now let's review. So which one? All of this. Yeah. So if you have pregnant woman, right, make sure you tell them about this. Now, now we're going to look at the disease affecting peripheral nerve. Peripheral nerve. Okay? Peripheral nerve. One of the disease affecting peripheral nerve. Look at here. This one. Leprosy. Leprosy, also called Hansen's disease, is the owner, the scientist discovered or really studied this disease, right? So now, everyone figure out what's the name of the microorganism cause leprosy? Mycobacterial leprae. Mycobacterial leprae. So which structure associated with mycobacterial leprae helps them to escape host immune response? Which structure? Macolic acid. So you need to remember. So you can, mycobacterial leprae is very similar as the bacteria we learned last quarter, mycobacterial tuberculosis. You guys remember tuberculosis? It's an intracellular bacterial infection, yes?
It's in the macrophages.
Yeah, high in macrophages because they have what? Acid-fast positive bacterial cell wall. What is acid-fast positive bacterial cell wall? They have a macolic acid. Macolic acid is a vaccine-like lipid outside the gram-positive bacterial cell wall. So the macolic acid inhibits what? Phagosome lysosome fuse. So they could also, the phagocytosis second-line defense stop. The intracellular infection. Okay? So why they go to the mycobacterial tuberculosis infect the lung, yes? This one, they don't like to, they like to hang out in the cooler temperature. They don't like to go to the lung, just hot, yes? So they like to infect the cell in the peripheral tissue, like cooler part of your body. You can imagine the finger digit and the tip of your nose, tip of your ear, they all cooler. Yes? So those is tissue get damaged. Right? So on the tissue, including the peripheral nerve, get damaged. So that's your symptom. Okay? So since this is intracellular infection, which type of immunity will be helpful? Tumor immunity or cell-mediated immunity? Cell-mediated immunity. Okay? So based on your immune system, some people, when you have infection, you have strong T helper 1 immune response. And some people, when you have infection, they have strong T helper 2 immune response. So depending on the default state, which pathway your immune system likely to activate first, then you have different symptoms. If you have a strong T helper 1 immune response, this T helper 1 can activate the cell immunity. You could have mild symptom, asymptomatic, or mild called tuberculosis leprosy. Tuberculosis leprosy is basically mild disease, not contagious. You could, some of the peripheral tissue, maybe the pigmentation pattern change or loss of sensation, but not very contagious. You can clear the way. However, if your immune system try to go activate T helper 2, guess what? You're going to develop lepromatous leprosy. What is lepromatous leprosy? You see? This is very contagious. The very, the tissue damage, disfiguring tissue damage. So back in the days, if you have those lepromatous leprosy, where you go? Back in the days, you what? No vaccine, but they have a bunch of antimicrobial antibiotics. So multi-drug antibiotics can be used to treat this. Yeah. Okay. So T helper 1, T helper 2. Let's look at them. So tuberculosis leprosy activated what? T helper 1. So it's mild disease, not contagious. You can even do skin test of type 4 hypersensitivity. If it's an activated T helper 2, guess what? You don't have lactobacillus. Yeah. And then connection with the immune system. So that's why, because T helper 1 started to increase what? Using interleukin 2, interferon gamma, right? So those type of cytokines promote to clear of the intracellular effects. T helper 2 producing interleukin 4, 5, including interleukin 10. Let's inhibit immune response. When you inhibit immune response, guess what? Microbes going to persist, multiply, cause damage. Yeah. Right. So how do we diagnose? How do we diagnose the leprosy? How do we diagnose? One thing is you look at it, we can see the symptom of the loose on your digit, which doctor? Yeah, right? In addition, you can confirm it and you get the T3, do the acid test, right? You do acid test, then you see acid test positive. Yeah. And then, where do you find the Mycobacterium leprae, this bacteria? Where do you find it? Where do you find it? Besides infect human, did you find it? You could look at this. I put in the name of the armadillos. Yeah, look at it. Yeah. You can read that. It's kind of fun. Hard to read. So the board could ask you, right? Armadillos, right? It's a natural reservoir for Mycobacterium leprae. Kind of fun fact, you think? Yes? Okay. Awesome. So you learn, and we have fun learning here. Yes?
Yes. I have an armadillo that comes to my house to eat our leftovers.
There you go. But infected.
All of them are? Every single one?
Not, I'm talking about not all. You have to have infected armadillo.
How do you know?
If they have infected armadillo, you might have human cases. So that's why you need to report. So if you're like in the hospital, they have somebody diagnosed with leprosy, you need to report. If they report and find out they have high case number, they probably should release the public statement about checking for the armadillo might be reservoir or something. You haven't heard, you can check with CDC, supposedly keep tracking of it, because all the disease reporting, they get data.
Will they ever die?
I don't think right now, there's a lot of leprosy in Georgia. So the armadillo in Georgia probably not infected.
So I don't have to put my time.
And unless you get leprosy, if you have a pet armadillo, you get leprosy, you probably do need to check your armadillo, yes. So, makes sense? Yeah. Okay, that's a good discussion. Now we really remember an armadillo, right? Okay. Right, so look at if you have leprosy, how many of the antibiotics could be used? Right, pyrazinamide, right, block the cellular transport. Right, you have isambutyl isoniazid. This one, guess what? Interfere with glycolic acid biosynthesis. You see, so it's multiple medication. You can also inhibit the ATP synthesis. You have multiple medication. So nowadays, the Mycobacterium leprae, once diagnosed, can be treated and cured. The key is you do need to do surveillance, make sure it's not spreading in the community or armadillo is not contaminated. Yeah. Okay. Next group we're going to look at is disease in the nervous system due to intoxication. We're going to look at some microorganisms produce exotoxin, which are neurotoxin. Okay, so now before we look at that, we learned endotoxin, exotoxin. You guys ready for review? So we're going to compare endotoxin versus ectotoxin, okay? Which toxin is made of lipid? Which toxin made of protein?
Exotoxins, lipids, and proteins are endo. No, just kidding.
So now we're going to review endotoxin versus exotoxin. Which toxin made of lipid?
Endo.
Endotoxin. Which toxin made of protein? Exotoxin. So exotoxin made of protein, okay? So now let's link that to the gram-positive versus gram-negative bacteria. Which group of bacteria release endotoxin? Gram-negative bacteria, because endotoxin is on the gram-negative bacteria cell wall, right? So let's review the gram-positive or gram-negative bacteria cell wall. So which bacteria cell wall is thick layer peptidoglycan? Gram-positive. Which bacteria cell wall is thin layer peptidoglycan and also have outer membrane lipid? Gram-negative bacteria cell wall. So the endotoxin is part of outer membrane lipid. Okay. So now let's look at the neurotoxin. So we're going to look at the two diseases, botulism and tetanus. Botulism, tetanus, both of these is disease in the nervous system due to what? Exotoxin. And they belong to which category? In the genus Clostridia. Let's review genus Clostridia based on this slide. Genus Clostridia, they are gram. Genus Clostridia, they are gram. Gram-positive, okay. Genus Clostridia, they are gram-positive. Shape? Shape? Bacilli. Yes? And where do you find the genus Clostridia? What's the reservoir? Genus Clostridia. The reservoir is in where? In the soil. Reservoir in the soil. How do they survive in the soil? They produce endospore. They produce endospore. And the genus Clostridia also is a obligate anaerobic. Obligate anaerobic. Right? Follow? Now, genus Clostridia is obligate anaerobic means what? Oxygen will kill them. Yes? So now, how do genus Clostridia, which is obligate anaerobic, survive the soil and the topsoil? There's a lot of oxygen. How do they survive in the oxygen environment? They form in what? Endospore. They form in endospore. So in anaerobic environment, that's the cell. Aerobic environment, endospore. Right? So now, let's review again. You see, you guys, final exam is very cumulative. Come from the micro 1 to anyway. So how do we kill the bacteria endospore? We have pasteurization, sanitization, antiseptic, disinfection, sterilization. How do we kill bacteria endospore? Sterilization. Yes. Okay, that's a review, yeah? So if we're going to ask questions about what I just reviewed, it's fair? Okay. Now, first disease we're going to look at is Clostridia botulinum. Right? So Clostridia botulinum, you see the bacillus, they form an endospore in the terminal end or subterminal end. So it looks like a tennis racket. Does it look like tennis racket? You guys agree this looks like tennis racket? Right? And the spore right there. So they discovered the 7 different botulism toxin. So what's the mode of action botulism toxin? You guys learned the neuromuscular junction structure? Have you known that? Acetylcholine activates skeletal muscle? Yes? Okay, guess what? Botulism toxin block acetylcholine release. So when you have acetylcholine cannot talk to the skeletal muscle, what's the symptom? Muscle weakness. Yes. So they figure out 7 different types botulism toxin. The therapeutically, you could use this toxin for what? Botox. Have you heard of Botox? Yeah. And in addition, this toxin is highly potent. So you could use as doctors, right? Yeah. So over here, so how do we prevent this botulism? botulinum. It's anaerobic bacteria forming endospore. How do they get to human?
Contaminated food.
What type of food? Contaminated canned food. Contaminated canned food. Because the endospore is associated with food, and the canning process you vacuum seal it. Once you vacuum seal the can, it's anaerobic environment. So this endospore goes in. Yes? So the symptom based on the dose. So it's a slight, a little bit of botulinum toxin. You might have a, like abdominal pain or like a constipation, they said. But if you have high concentration, if you paralyze the diaphragm, the patient could have to rely on the respirator. You see? Sometimes it really depends on the concentration. Now, besides like contaminated canned food, home canning, that's a risk factor. Another group at risk for botulism is infant botulism, due to the infant eat honey. Have you heard of the don't eat baby honey? Yes, because the honey obviously comes from natural environment. It could have endospore contaminated. But when the baby gets older, they have a healthy probiotic in the gut, so they can inhibit that. But for babies, they do not have a really complicated gut microbiota, so you have even one or two endospore of really large one, you could have. Yeah. And then, so how do we prevent the canned food from like botulism? What do you do? Commercial canning. Commercial canning, you have to use you treat the food with what? Heat with pressure. You have to autoclave a little bit. Kind of, because 100 degrees, you cannot kill the endospore. You have to apply the pressure, right? So for home canning, you might need to use a pressure cooker before you canning it. Another thing you can do is if the home canning food is acidic, for example, tomato sauce, those types of things, you do not need to worry about botulism. But if it's pH neutral, like beans or potatoes, those types of things, if you do not follow the procedure, that's a risk for botulism. Okay? So, right, there's food intoxication, Staphylococcus aureus, salt tolerance associated with salty food contamination, and canned food, a risk for botulism. Now, easy, right? Good. Right, okay, awesome. The next one we're going to look at is what? Tetanus. What's the name of bacteria called tetanus? Clostridia tetani, right? Clostridia tetani from an endospore again. See the, like tetanus racket? So, do you know how, in general, how do you get the tetanus? Typically rusty nail, yes, rusty nail in the soil. So rusty nail, one thing is soil, they could have bacteria endospore. Another thing is once you step on a rusty nail, they produce what? Deep wound. When they have deep punctured wound, the deep end, they could get to, that could be an anaerobic pocket because it's anaerobic bacteria. So if it's a superficial wound, you do not need to worry about tetanus because the oxygen will inhibit it. But you have deep wound, that's the risk. Okay? So you can see now, the tetanus also leads to neurotoxin. Let's look at the toxin, right? So the toxin, the tetanospasmin, is the toxin inhibits the nervous system, toxin, skeletal muscle. So what happens is, you see, any type of movement, you have group muscle constrict, another group muscle is relaxed, yes? So tetanospasmin, what happens is make all the muscle constrict. When you have all the muscle constrict, what's the symptom? Yeah, locked jaw, or you can see the infant botulism. See, all the muscle constrict. Okay, now, right. So this could be deadly. So if you look at this picture or probably through, not through parents. Typically it's a unsanitized childbirth. So if you have a contaminated, like dirty scissor cut umbilical cord, it could that's it. For the adult, it depends on the dose. If you have like a low concentration, maybe part of just rigid, you lock jaw, those types of things. But if it's high concentration, you could have all the muscle constricted out. Right? So that's tetanus. How do we prevent tetanus? Yes, vaccination. And the tetanus vaccine need to have booster shot every 10 years. Get a booster shot. Okay. And if you can see the treatment plan, right? So what if you refuse to get tetanus vaccine? What do we do with people anti-vaccine? Then I don't want to get tetanus vaccine. How do you help? Metronidazole, metronidazole to kill the anaerobic bacteria. In addition, you have to do what? Passive immunity. You give them anti-serum, neutralize the tetanus vaccine. The good news is you have anti-serum available. So use the antibody neutralize toxin, using metronidazole, killing the bacteria, it can help. In addition, another thing we could do is, you guys remember catalyst test? Catalyst test we learned last quarter. Hydrogen peroxide, catalyst make oxygen bubble. So if you have deep puncture wound, if you pour the hydrogen peroxide over, you generate a lot of oxygen bubble. So the oxygen bubble will prevent Clostridium contaminant in those four germs. Very good. Make sense? Now, so the neurotoxin, right? Neurotoxin in here, bacteria produce neurotoxin. Example is what? Botulism toxin and tetanus. Right? So that's an example of some bacteria infection in the nervous system. Any question? That's no question. Now we're going to look at what? Viral disease in the nervous system. Okay, viral freedom. So let's differentiate between bacteria and virus. So what's the difference between bacteria and virus? Yes, virus obligate parasite. Yes, what else? Virus, they are not composed of cell. They are not living. They are acellular. Bacteria is composed of cell, composed of prokaryotic cell. Yes? Okay. So now, what is virus made of? So not composed of cell. What's it composed of? They have a DNA or, alright, not both. They have one type of nucleic acid, either DNA or what else they have? Some of them have lipid envelope, some not. So if they do have envelope, we call them envelope virus. If they do not have envelope, we call them naked or non-envelope virus. Yes? What else they have? They have a protein coat called what? Capsid. Capsid, right? The protein coat. Yes? Okay. Awesome. So that's review of both virus. And the prion is made of protein. Yes? So they could cause meningitis, encephalitis with no simple flu. Okay? So, one real problem common like viral meningitis is come from the enterovirus family. So the enterovirus family, the coronavirus. So these group of virus, they have a naked. There's an RNA virus, naked, not envelope. So now, what's the difference between the envelope and non-envelope virus? Yes. So which group of virus we could prevent with washing hands? Envelope or non-envelope? Envelope virus, because envelope virus have lipid envelope. Use soap, use alcohol, you can destroy the lipid. Now, non-envelope virus, the surface is what? Protein. in general, okay, give you a trick. In general, if the virus portal entry is fecal oral, they get what type of structure? Non-envelope. Because non-envelope virus, they could survive the mucous membrane, survive the stomach acid, then go to GI tract cause problem. If it's envelope virus, when you ingest it, the whole thing will be destroyed. The lipid will be destroyed. Make sense? So now you can see the common childhood viral meningitis could come from Coxsackie virus, A and B echovirus. Those two viruses are mild, so it depends on the patient. So some cases just have a called stomach flu type symptom. For Coxsackie virus, basically vomiting, fever, and also might have rash on the hand or the mouth rash. So mild. But for some kids, if you have weak immune system, the virus could cause mild viral meningitis. Make sense? Okay. So how do they get it? It's come from a fecal oral contaminated food ingestion. Yes? Okay, they're not virus. So if you have viral meningitis, how do you diagnose viral meningitis? How do you diagnose viral meningitis?
Spinal tap.
Spinal tap. And then you look at what? You look at the glucose concentration. If viral meningitis, glucose level is what? Normal. And increase the lymphocyte. See? Right? And then, oh, the one is here. I think we have a, you guys look at the discussion, topic discussion. This week we have Iron Lung story.
Yes.
Yeah, you like that? There's one guy, I think he lived like 70-some, right? He just died recently, within like 2-3 years, right? He was booked, lived a whole life, the complete life, right? Went to college, he didn't have a job, really good story, yes. Complete poliovirus infection, okay? So, how do you get a poliovirus infection? Fecal oral contaminated food water. So, what's the structure of poliovirus? No, yes, yes. And then, so obviously, once you ingest the contaminated food water, virus will access the GI tract. And then the virus can, from there, it can access blood vessels. And then they can, from blood vessels, they can also access nervous system. So the poliomyelitis, now you guys learned the nervous system? Have you learned the spinal cord, structure of spinal cord? Anterior horn, posterior horn, have you heard of? Not yet? Not yet. Oh, okay. So maybe when you learn it, you can make connections, right? So this is the cross-section of spinal cord, right? So the poliovirus could access the motor neuron, anterior horn. So the anterior horn is motor neuron. So if the motor neuron is affected, then what's the problem? Paralysis. Yes? Make sense? So the anterior horn, motor neurons, you affect your motility, yes? Make sense? Okay, awesome. So when you do learn that chapter, making connection with poliovirus, okay? So now, how do we prevent nowadays? How do we prevent polio? Vaccination, right? Or you just make sure like only water, cookie food, yes? And in fact, the human, the fecal sample treated? Yeah. Now, the vaccine worked or not? In America. Look at the case. Vaccine worked or not? It worked, yes? Right? You can see in the 40s, right? That's where the big epidemic in America. Worldwide. What about worldwide? Vaccine worked or not worldwide? You see, right? Worldwide in the 2002 versus 2016. See, right? So the vaccination worked, right? Not including, not only reduce the case number in America, but also worldwide, global. Occasionally, you're going to hear the polio outbreak in the community, in the war zone, or the refugee camp. Sometimes it's because the fecal oral contaminants, unsanitized living condition, lack of access to vaccine, could have this disease could come back. virus, right? Contaminated food, water coming in, GI tract, then they're going to access the GI tract through blood vessel, through nerve, they're going to access what? Spinal cord. So they're going to infect and multiply in the anterior floor of the spinal cord. That's the motor process. Okay. So depending on which location, different muscles get affected. Most common symptoms like that. Then, yes, lower limb. Right. Now, in America, guess what? The diaphragm muscle is affected. So when you have diaphragm muscles affected, guess what? In order to survive, you need iron bond. Right? Iron bond is a piece of metal. Okay? So you like the story about polio?
Yeah.
Yeah? Interesting, yes? Okay. So the next disease, you guys want to break or you want to continue? Now break, okay. We'll come back at 405, yes? 405. Gonna look at rabies when we come back. Make sure to come back on time, okay? 405.
like about where it is?
Yeah, it was the bar.
The Oxford. Yeah, I did a side profile for that too. I feel like I should have used a better picture. I had so many nice stuff because I might redo like labeling or something. Yeah. And it's hard to be perfect because you're. I wish I had iPads sooner because I would have. But it is what it is. Next term I'll be doing it right away. We didn't start. Yeah, I was worried about angles, but then I realized a lot of the papers indicated either posterior or anterior. So I'm like, is there a reason for my angles or not? I don't know. On his YouTube videos, there's a reasoning behind the angles. If you're just going by other labeling, there's no that. It's kind of your preference how many you need. Actually, if you need every little one, then the angle matters. But if I don't know. I feel like I should. The fetal skull, that was the hardest one because some of the sutures, like is it the fontanelle or is it the it's the fontanelle is not a suture, right? So then it's something else. It's the anterior line. So that was the first part. I wish, are we allowed to include stuff not on the list and then, or do we just go back? That's what I thought. I found 5 vertebrae and this one. The spine, it's irregular dogs. Irregular dogs. It's like flat, short dogs. That's right. Yeah, the first thing I did was I knew that was happening, and then I did the compact bone, so many parts there, and then the types of bones, right? This part? Oh, no, yeah.
Yeah.
Scapula. That one has a lot. That one is one that you kind of have to turn. I don't know. I just put the anterior and posterior. That's it. If I don't have enough, then I'll look. But it's exciting. It's exciting. Doing up all this stuff. Like, it reminds us what we're doing.
We got one.
Do you want to stretch your legs?
Yeah.
Do you want to get some sunshine?
We had a rabid coyote in my backyard one time.
Oh, really?
Oh man, not too long ago. There was we were in Sandy Springs,
Yeah.
and a raccoon showed up. Foam in the mouth was. We had to call animal control to come get it, and they went to go get it.
Okay, you guys okay?
Oh yeah, no, we were fine. It was just we had to see the middle of Metro Atlanta, Georgia, just this rabid animal walking by.
We're gonna talk about it.
We wouldn't go get it because it wasn't technically. There was was a weekend, first of all, and they said technically it was too dangerous for them to come yet. I don't get it. That's their job.
Okay. Yeah.
That's crazy.
Try to go hiking?
Hiking? Sounds fun.
Yeah. What about you?
I'm going to Lake Norman for Memorial Day.
Oh, holiday.
Yeah.
Oh, party?
Not necessarily sorry, see some friends.
Okay, good.
Yeah.
Yeah.
Might get in some trouble.
Yeah, well, just, well, whatever you eat, we learn a lot about microbial effects on social food, the drink, the
I'll be
animal,
safe.
yeah. We go
safe.
camping, yeah.
I know what to do now.
I know.
That's not the brain bacteria.
Yeah, if you swimming, watching what? When you eat we're gonna look at tomorrow. Yeah.
Yeah.
If you eat food, make sure it's good. Yeah. If you can't see, watch out wild animal, raise these out. They enjoy.
I was gonna say, wow, animals. I was more worried about hiking.
Well, hiking, that's the, I get my dog.
What dog you have?
I have tiny, Jack Russell mix. Very high energy, it's a good hiking dog. So we have to walk it to get tired.
Awesome.
But anyway, welcome back. Ready to continue the wiring and testing your nervous system? Yeah? Okay. So now we're going to look at retrovirus. First of all, let's look at picture of retrovirus. Describe structure of retrovirus, right? DNA or RNA virus? Retrovirus, DNA or RNA virus?
RNA.
RNA virus, right? Retrovirus, RNA virus, single strand RNA, right? Negative strand. What about the envelope? Envelope or non-envelope? Envelope virus. So envelope virus. So how do you get the retroviral infection?
Bite from an animal.
Affect the animal product will bite. Yes. So this is not a disease. So shape of a retroviral shape look like a bullet. You guys agree this look like a bullet? So if you see the question like a bullet shape, envelope RNA virus, we're talking about which virus? Retrovirus. Yes? Okay. So now, this is zoonotic disease, so it's an animal bite, scratch, or inhalation. Okay? Now, in Georgia, so it's a wild animal, right? In Georgia, which type of animal more likely carry retrovirus? Raccoons, right? So now, the control of retrovirus. So how do we control retrovirus? To prevent the human infection. So if you have a dog or cat, what you need to do?
Vaccinate.
Vaccine, right? So animal vaccine is strongly recommended, but the human vaccine is not. Okay? So the retrovirus prevention is to prevent the animal infection with vaccination. Another thing is how do we get the wild animal vaccinated? So ideally, the public health, what they trying to do is they use the airplane to drop the rabies vaccine. They mix the rabies vaccine with the food. Then it's to go to the mountain, talk about the hiking, right? You can wild animal, right? So what they do is they supposedly have they're going to use airplane to deliver the vaccine in the mountain. So hopefully the wild animal will eat those vaccines. That way you can vaccinate the wild animal. So when humans go hiking, take the dogs, so they won't have encounter the wild rabies animals. You think? Right? new story, right? So ideally, what they're supposed to do is they periodically drop the rabies vaccine with food, just delivering it in the wild. So the wild
But
animal-
that raises the question: if an animal is in an urban environment, what happens then? Because that raccoon was in the middle of Sandy Springs.
That's why it's if the public health is not doing a good job, then you have rabies vaccine end up in urban life. And right now they cut in the public health department, right? Save money. So if they do not really, if they supposedly deliver the rabies vaccine, so that way the urban environment you won't encounter rabies animals. Yeah, make sense?
Yes.
So once they already get in there, then this is where a human now. What if you encounter really wild animal and get bite? What do you want to do? Yes? Okay, so let's watch this slide. So this slide, you see, the virus enter. It has peripheral nerve, so the virus can travel with the pathway for the virus. Rabies virus go to brain through peripheral nerve. Just travel along the peripheral nerve, go upwards, all the way to the brain. So this is the disease is very slow, progressively getting worse. Okay, so in initial stage it will be like a flu-like symptom, this part. As the virus multiply, affect more peripheral nerve, then you're gonna have more symptoms. Now, how do we know the wild animal bite you have rabies virus? How do you know?
Coming in the mouth?
If the wild animal bite you, they could it's like, oh, I'm wondering if I got animal bite. Do I have rabies or not, right? What you do is you grab that animal. Grab the animal, what you going to do?
You have to inspect its brain.
Yeah, do the brain biopsy. That's why the animal needs to be euthanized. So that's why you have policy, if the other animal, including your dog, bite other humans, you might want to euthanize the animal. Just check the brain to see if the virus in the infected animal. So how do they check? Another thing I want you to remember is if they discover Negri body in the neuron, that means that the brain tissue is infected with rabies. Another thing you can do is immunofluorescence. You can use antibody to fluorescence stain to see that the Negri body is smaller. In fact, in human, initial stage, prodromal stage, not specific symptom, right? It's tired, you have flu-like symptom. Fever stage, this is where you start to have normal activities, including what? Hydrophobia. So the unique symptom is when you get in the fever stage, patient they kind of thirsty, they need to drink water, but they're afraid of water, they can't drink it. They need water, hydrated, but they can't drink it. In that stage, it's kind of too late to treat it. And then you have comorbid. Right? So now, how do we deal with human case? As soon as you get a suspected animal bite, you need to grab that animal because they will euthanize it and check the animal brain. In the meantime, human need to do two things. One is you have anti-serum neutralized, so passive immunity. You have anti-serum neutralized rabies virus. In the meantime, you're going to get rabies vaccine. Rabies vaccine several doses. So now, vaccination in general is for prevention, right? But the rabies vaccine in humans for what? Treatment. You see? So most vaccine in human, we use it before you can encounter microorganisms, yes, for prevention. But the one exception is rabies vaccine is used for treatment. So once a human exposed to the suspected animals, including inhalation, if you have close encounter with the infected animal, you probably need to check, even though they do not bite the inhalation. Yeah. So you need immediately taken passive immunity, anti-serum. Inhalation, vaccine. Okay? So, and the animal need to be euthanized and check the brain biopsy. Let's look at this question. Which one, everyone?
B.
Which one? B. Rabies. Yes. Awesome. Now, next we're going to look at is measles, we're talking about, we already talked about this virus in the last quarter during the respiratory tract vaccine. Yes, we're going to bring back here again because the infected individual after recover from measles, they could have what type of disease in the nervous system, everyone? Subacute sclerosing pain encephalitis. So SSPE, subacute sclerosing pain encephalitis, could occur 5 to 10 years later after patient survive or recover from it. Yes, so that's why measles vaccine is one of the vaccine very important to prevent the measles outbreak. Okay, so on the current topic in Texas, right? They have how many cases now? Like over 100? Yeah, over 100. They have a couple kids died. So viral meningitis leads to death during the acute stage, and then you have subacute sclerosing pain. Okay, so now if you encounter this measles viral infection, first of all, let's describe the virus structure. Measles virus, measles disease, also called what? Rubiola. So the virus belongs to the mobility virus family. Mobility virus family. And the structure is what? Enveloped RNA virus. Right? So enveloped RNA virus, how do you get it? Port of entry is what? Port of entry is what? Yeah, inhalation. Respiratory droplet. Inhalation. So how do you prevent this measles virus if you live in the area there's outbreak? One thing you can do is what? You can get a vaccine. Another thing you can do is what? Wear a mask. Washing hands. Because it's envelope virus, washing hands could be helpful. Wear a mask, right? Information, wear a mask could be helpful, yes, plus vaccines. So now, if the patient have measles virus, how do you diagnose them? Rash, and also the complex spot. So if you open up in the oral cavity, you have a complex spot, white spot design in the oral cavity mucous membrane. So that's the classic symptom of measles. Right? Now, with measles, I do want to point out a couple signs. Right? So we just learned rabies, measles, we learned tuberculosis previously, right? So let's look at this slide. Which disease is most deadly? Rabies. So the death rate is 99.9% if you do not get treated immediately. Okay? So which disease have a lot of asymptomatic individuals? Tuberculosis, right? So the measles in the middle, right? Measles, you have a few asymptomatic cases, but not a lot. And the death rate is not as high as rabies, but the death rate could occur, yes? Right? Now, let's look at this data. Based on the infection dose, what is infection dose? Infection dose we learned previously. I'm kind of review the micro one a little bit, yes? Infection dose is talking about how many microorganisms could lead to the disease in a human, right? So, based on infection dose, which one is the most transmissible? Measles. How many? Just one. You see how contagious? Right? So you see, just one variant could lead to disease. Now, based on the basic reproductive number, right? So if you have one patient with measles disease, if you do nothing, you do not wear masks, you do not do any social distance and washing hands, from one measles patient, it could lead to how many additional cases? 12 to 18. You see? See? Right? So very contagious. So that's why in Texas, every time they have a measles outbreak, you really get worried because it spread quickly. Right? Basic reproductive. Yes. Okay. Now, next we're going to switch gears. We're still looking at viral meningitis and encephalitis. The next category, look at here. First of all, you see the diseases, viral meningitis, encephalitis come from Easter equine encephalitis, right? And the Western equine encephalitis, and the Zealand equine encephalitis, St. Louis encephalitis, West Nile, California encephalitis. rotavirus encephalitis is a serious disease, yes? They belong to the family of rotavirus, flavivirus, rotavirus. So what's the structure of those groups? So what type of nucleic acid they have? They have RNA, right? RNA virus. You see? So they all envelope RNA virus. Some of them plus strand, some minus strand. Envelope single stranded RNA virus. See? Right? So that's why it's not the it's envelope of RNA virus. So it's not spread by contaminated food water. Yes? It's a direct infection mosquito bite. Right? So this group of virus, another name called what? Arbovirus. So arbovirus is talking about the virus, vectors, arthropod. So they kind of include a variety of viruses. The vectors, the arthropod, could be mosquito, could be ticks, could be mice. That's arbovirus. So once they go in, they could cause meningitis and encephalitis. Yes? Now, what's the symptom of meningitis and encephalitis? You guys know? What's the general symptom of encephalitis and meningitis? You guys know? Sometimes with high fever, right? And yeah, enlarged lymph node, high fever, stiff neck, vomiting, because depending on which part of the brain is affected, anesthesia, yes, headache, all that. And over here, you're talking about arbovirus life cycle, yes, mosquito, the reservoir could be horse, small mammals, bird. How do we prevent this group of viral infections? Yeah, bug spray. So you see the endemic region in tropical area. Tropical area, one thing is you have all bacteria, yes. Bacteria, reservoir animals, yes. So, okay. Well, there you go. So you have basically avoid mosquito bite, yes, and also mosquito bite the human in the evening hour. So the evening hour when you go out, yeah. Awesome. So now we're going to look at sample for viral infection in America. West Nile virus. So West Nile virus. So West Nile virus, what's the structure of West Nile virus? Everyone see?
Stranded RNAs.
Envelope, single strand RNA. Yeah, envelope virus, right? RNA virus, envelope virus. So who are the reservoir hosts? Yeah, you see the bird, the horse, right? Who's the vector? How do human get it? Who's at risk for serious symptoms? At risk population for West Nile virus is seniors and children. Immunocompromised. You could have encephalitis. How do you diagnose West Nile virus? Diagnose, you could use enzyme-linked ELISA, antibody antigen neutralization, or you can use some, microscope. Look at it. Now, fun fact. This one kind of interesting. So which year in America the West Nile virus first saw up? Which year? 1999. In which state? New York. How did they get there? Airplane. Yeah, the New York, that's the international airport, right? Then, so that's the first year that people noticed that. So now, which year? By which year? How many years later? The West Nile virus saw up in all states in America. You see, 4 years later, you see how quickly the virus spread? Now this disease is established in America. Yeah. So now you see the monitor. So to be aware of it, if you have time travel, let's say you can have time travel to 1999, what are you going to do? Right? You can stop that plane. You can do a time travel movie, right? Or whoever get that disease get quarantined. Don't go out there and get mosquito bite. You see? Right? You mainly so you need to prevent the virus to spread to mosquito. Once the mosquito get infected, they can spread to more people. So from public health perspective, yes. So the airplane, the Who's at risk for severe symptom of Zika virus? Yeah, pregnant woman, because the baby could have microcephaly, had baby, right? But for the immunocompromised, non-pregnant woman, you could also have virus, like malaria, so you have some kind of muscle pain, fever, including meningitis. Okay. Now, next group. So this Zika virus belongs to what? Flavivirus, right? Flavivirus. So it's an enveloped Austrian. So look at it. So this one, we'll look at how many viruses in the flavivirus family. How many viruses? You see? That's Nile virus, Zika virus, St. Louis, encephalitis, dengue virus, yellow fever, right? Including tick-borne encephalitis, virus. So this RNA virus, the RNA is very unstable. So the virus in this family have high mutation rate. If you look at this phylogenetic tree, you see, right? So this is more Asian, a little bit older virus. Over here, you can see the tick-borne encephalitis relatively recently along. So these group have high mutation rate. You see how many viruses still showing up, new virus still showing up. Make sense? So now, right? So over here, we're just going to look at the different angles. For the RNA virus, could be plus strand, could be minus strand. Yes? Could be envelope, could be naked. So the flavivirus is what? Plus strand RNA virus. The Ebola virus we learned last week. Yes? Ebola virus belongs to the flavivirus. That's minus strand. See? Now, we're going to making connection. One of virus we're going to look at tick-borne virus infection, tick-borne encephalitis, and virus. So we're going to use virus as an example, making connection with our immune system. Right? So virus could lead to viral encephalitis, can be serious disease. So early stage, right? How do we detect virus in early stage? We check for what? Viral antigen test, like COVID-19 antigen test, right? You check the variant. So, and then a couple weeks later, antibody test for early stage of infection, we check for what? We check for antibody, IgM. So if it's chronic infection, which type of antibody show up? IgG, see? Right? Now, look at how many of microbial infection associated with tick. We learned a bunch of bacterial infection last week, yes? Right? Okay. So now we're going to use this slide after this question. Right? The key is right here. So the infected tick, right, we transfer a bunch of bacterial infection by anti-disease, Rocky Mountain fever, right? It's also viral infection, yes. Which one? Okay. Caused by virus. Which one? Which one? K, yes, K, yes. Yeah. Okay. Now, still based on previous slide, let's answer this question.
B.
That would be what?
B.
B. There you go. Good job. B. Okay, so we have fun. Yeah. So another group of viruses could cause disease in the nervous system. Guess what? Herpesvirus. So let's review herpesvirus structure. What do we know about herpesvirus? DNA or RNA virus? Herpesvirus is DNA virus. DNA virus. Envelope or naked? Envelope the DNA virus. Okay? So So herpes unfolded DNA virus. What type of disease herpes typically cause? Acute, chronic, or latent? Latent. And once you have herpes virus, you carry forever, right? You can't have a current infection. How does herpes virus relate to the disease in the nervous system? Right? See the herpes virus 1 and 2. The latent in where? Yeah. Yes, you know. Yeah, good job. What about the chickenpox shingles? Where do they, varicella zoster virus? Where do they get latent? You see? So the latent in the nervous system. Right? So let's look at herpes virus 1, 2. Right? Herpes virus 1, latent in where? Hygiene there. Yes? And herpes virus 2, latent in where? Seasonal gland. See? Right? Now, and then the varicella zoster virus. Where do they get the latencies in where? In the posterior loop of ganglion. See? That's the sensory loop. Right? So you see the chickenpox and shingle we already talked about, we just reviewed, right? So how do you get chickenpox? Primary infection is what? Inhalation. Then primary disease is the pox, right? That's Slovenia, right? Everywhere, right? Fever, very contagious. By the time you have shingles, guess what? The disease reflects where the virus is hiding. Make sense? How do we, right? So the shingles, so the primary chickenpox disease everywhere, pox. And the shingles reflect the virus where they've been hiding lately. Yeah. So how do we prevent the chickenpox shingles?
Vaccine.
Right, vaccination. Now, if you refuse to get vaccination, what you can do? All the herpes virus, you have anti-venom. Yes? Yeah. That one too. Yes. Okay. So now, another viral infection affects the nervous system is the JC virus, aggressive multifocal leukoencephalitis. And we have this one. This disease only occurs among immunocompromised, severely immunocompromised 8 patients. Right? So the virus, polyomal virus, what's the structure of the JC virus? Non-monopolar DNA virus. Okay? So only occurs for immunocompromised patient, aggressive multifocal leukoencephalitis is dead. Okay? Who carry it? You. Now, lastly, we're going to look at this acellular particle. We got prions. So what do you know? This is a review. Prions review. Okay? So prion made of what? Protein only. So the prion, you have the cellular normal prion. Where do you find the cellular normal prion? In the brain. So the infectious prion. So what's the difference between the infectious prion versus the normal cellular prion? Is what? The infectious prion have more beta C. You guys learned alpha helical beta C in the biochem class, protein secondary structure, yes? So the folding. So when the infectious prion is they change the folding from alpha helix to beta C. When they change the folding, guess what? They have additional characteristics. One thing they have enzyme activity. They can convert in a normal prion into a normal. Another thing is they are highly resistant to all kinds of treatment. So how do you get rid of the infectious prion? How do you get rid of the infectious prion? You have to literally extra sterilization. If you autoclave, you have to autoclave for 4 hours instead of 30 minutes. Or you can just literally incinerate. So you infected the prion disease in the animal. How do you treat the body? The carcass, you have to burn it. You literally need to burn it. You can't bury it on the ground. You have to burn it. Yes. Right? So how do these infectious prion multiply? How do they multiply? How do they multiply? They are like a coat, lead. You guys know the they do? You know cult? C-U-L-T? So how do the cult leader recruit more members? They brainwash. Yeah, brainwash. Yes. So the infectious prion go to your brain, convert all the normal prion into abnormal protein. When the normal prion convert into abnormal protein, what your brain look like? Sponge-like. You see the hole in your brain. Right? So you can like do a biopsy or you can do imaging, you can see the abnormal brain. So now, since we have normal cellular prions, so sometimes the prion disease in human could be genetic, inherited. Could be sporadic mutation, could be inherited. So this one you just have to manage. But public health could control what? Infectious prions. Infectious prions coming from what? How do humans get infectious prions? Come from contaminated animal meat. Remember the mad cow disease associated with beef products back in the days? Yes. So public health could control the spread of the infectious prions. So now, over here you can see how this prion disease is very common among our animals as well. Yeah. Right. So that's why if you have sheep or cows, you have prion disease, you have vitamin burn. If you throw the body out there, other animal eating it, you spread it in our animal. Eventually, it will come back to human. You see? Right? So now, can you answer this question? Infectious prion disease is called what? Variant Creutzfeldt-Jakob disease. So Creutzfeldt-Jakob disease probably comes from the either scientists with treatment disease or patient, right? So why the infectious form called variant Creutzfeldt-Jakob disease? Because the normal Creutzfeldt-Jakob disease is a genetic disorder, and the variant Creutzfeldt-Jakob disease due to infected animal. Make sense? Right? Okay. So if you look at this table, kind of, if you cannibalism also occurred due to what cannibalism. So back in human history, they had a famine. In human history, the famine, during the famine, people started eating people. Then they discovered food. Yeah. Right? And then the symptoms associated with brain damage eventually yes. On the diagnosis, you can use the X-ray or imaging or the brain biopsy. And the treatment, there's no cure, you just manage it. Right? So that's prion disease. Any questions? If you don't have questions, we can just look at the 1 or 2 eukaryotic infection before we go. How about that?
All right.
Okay, just 1 or 2, not a lot, but we have a lot of there's lots of microbial infections in the nervous system, okay? So you see the eukaryotes. Who are eukaryotes? Fungi. We are eukaryotes, including what? The fungi, the protozoa, the worms, yes? So this is a list of disease we're going to look at, okay? First, we're going to look at fungal infection. So the fungal infection in the brain could be due to what? Intoxication. So fungal infection in the brain could be due to toxin intoxication. Some of the toxins have this hallucinogenic effect people actually like. That's what? Yeah. Right? But a high dose could cause brain damage, yes. Some of fungal infections due to what? Fungal pneumonia. Remember we learned much of fungal pneumonia in last quarter? Inflammation, histoplasmosis, clostridium. Oh, funny. You can do the literature. Drug, you use too much, not good. Don't do any drug. That's my advice. Anything influencing brain, you have to do some research on that, right? You can follow up the research. So the really high dose frequent, they do have some literature. You can search PubMed and evidence. Make sense? Yeah, evidence-based. You go check with some literature because America now is legalized marijuana, all that stuff. So now we have a lot of evidence now. Coming from, and it just recently, it's like a couple years, they legalized it.
For medicinal use
like open up this? Yeah. You guys know it, right? Right? But basically have a lot of documents now. Since more people is using it and also to open about it, right? The CPD stores on the street. So now you actually accumulated evidence. You have more of actual evidence to show you so what's the effect of this medication, long-term use, what's against society. Oh, that's really bad. Only several years ago, the teenager vapor and all that stuff, they have all kind of problems. Yes. So my advice is to just eat healthy, go exercise, go hiking, and right? Okay, good sleep. That's all you need. You don't need anything else. Or something. Yeah. Say no to drugs. Okay. Right. So fungal pneumonia could lead to fungal meningitis. Okay, so we're going to look at a couple. So one's poisonous mushroom. Another one is moldy bread, moldy wheat. So if you use moldy bread, moldy wheat, you can have what type of toxin? Ergotis. So ergotis, when you see the weeds, right? Barley, mold grow. If you use this barley to making bread, guess what? You could have a convulsive ergotis. Convulsive ergotis back in the days leads to the witch hunt. Have you heard of story witch hunt? Actually, in history, killing like 18 women, they think they are witch because they eat this poisonous bread. They start at weird, neurotoxin. Yeah, right? You heard of that? Yes. Now they look back at history. Say, oh, in that year, there's a lot of rain, so a lot of barley is moldy barley. People still making bread. That leads to like a group of people just kind of act like witch. They got killed. How about that? Right? But anyway, so another type of ergotis due to what? Gangrene. Gangrene is going to be tissue damage and necrosis and that stuff. Yeah. Okay, that's intoxication. Now, with fungal pneumonia leads to fungal meningitis. Guess what? You guys still remember? Histoplasmosis, blastomycosis, coccidiodine mycosis, paracoccidiodine mycosis. We learned that. You guys remember? Right? Now, look at this map. This what type of disease? Epidemic, pandemic, or endemic? Endemic disease. How do you get this group of fungal infection? How do you get it? So the reservoir is in the soil. How do you get it? Inhalation. Fungi. They produce what? Fungal spore. Inhalation of fungal spore. So the soil have those type of mold. And also they have thermal dimorphism. You guys still remember thermal dimorphism? Means what? Thermal dimorphism means a dimorph means two form. Thermal based on temperature. So this mold grow in two forms based on temperature. Yeast form in where? In human. Mold form in where? In the soil. So in the soil, mold form mycelial hypha produces spore. Once a human inhale a spore, first organ affected is what? Lungs. The human lung temperature is high. They're going to produce single cell yeast like. And then one of them, fungal pneumonia called coccidiodine mycosis. Let's see. They can easily spread into fungal meningitis. So coccidiodine mycosis is very serious. So this one, primary infection is what? Fungal pneumonia. And then leads to what? Fungal meningitis. Okay? So where do you find those type of mold? Coccidioides. Where do you find it, everyone? It's very significant in America. You could be tested in the board. Where do you find it? Everyone is right. Southwestern U.S. Which state? Southwestern U.S. Yeah, the desert outline. Right? Arizona, California, Nevada over there. Yes? Okay. So now let's look at some more dimorphism. After this, we're going to have break. Now we're going to go. How about that? So now, in the soil, which form? In the soil, which form? media get in human lung, forming which form? Spherules. So if you see the spherules is a structure inside the single cell, we call endospore, but it's not bacterial endospore, it's yeast. So yeast, single cell yeast inside where? Spherule. So the sphere in human lung, and they can decimate to the brain. Got it? Okay. So you guys want to look at another one, or you want to?
Do we have time for another one?
You want another one?
Do we have time for another one?
2:45. How about just do knowledge check and quickly? Let's have 5 minutes, right? You guys need time to do knowledge check.